Good morning. My name is Dr. Navdeep, Assistant Professor in the Department of Physical Science. So today I am going to introduce the topic of Spermatogenesis and Oogenesis. These both are in the subject of zoology in the category of developmental biology. So spermatogenesis and oogenesis are actually the formation of the sperms and ovum. So first we discuss spermatogenesis. Spermatogenesis, in spermatogenesis we have spermatogonium type A cells. So these all are cells formed in the seminiferous tubules of the testis. So if we look into the microscope, so we have many different kinds of cells in particular in the testis. So all these cells are actually the different stages in the spermatogenesis process. So here these stages. So first spermatogonium. It has 44 plus X plus Y chromosomes. So this is the chromosome number in our cell. All the chromosome numbers are fixed in particular species. So first spermatogonium type A undergoes mitosis division. Mitosis and meiosis are both the type of cell division. Mitosis is the equational division, means chromosome number remains the same after the cell division. But in the case of meiosis they have two types. First meiotic division and second meiotic division. First meiotic division is the reductional division, means chromosome numbers reduces to the half the chromosome numbers. Means if we have 44 plus X, then we only remains 22 plus X or 22 plus Y. And the second meiotic division is also as the mitosis stage, means chromosome number remains the same after the cell division. So here spermatogonium after undergoes mitosis give rise to spermatogonium type B, means 44 plus X plus Y chromosomes. Then after some time spermatogonium type B or A undergoes first meiotic division. First meiotic division produces two primary spermatocytes. Primary spermatocytes reduces to the secondary spermatocytes. Now we have 44 plus X plus Y reduces to 22 plus X and 22 plus Y. So these both are the secondary spermatocytes. Then these secondary spermatocytes undergo second meiotic division. In the category of second meiotic division, chromosome numbers remain same, but it reduces to the two daughter cell. We have now overall this process, the result is this process reveal that one spermatocyte produces four spermatids. And after the development of spermatids, we have now 4 spermatozoa after the maturation. Right. So second process is the oogenesis. Female gonad is, we already know that, ovum, and ovum have also the ovary. We also have the different kinds of cells. During this process, both the oogenesis and spermatogenesis process almost are same. Here we have some differences only, but similarity is that similarly oogonium produces to primary oocyte, then undergoes first meiotic division, produces to secondary oocyte, and first polar body here we have. In this spermatogenesis, one spermatocyte, spermatogonium produces to 4 spermatids. But in the oogenesis, one oogonium produces to only 1 ovum instead of 4. Right? Here is a difference. And another difference is that when in case of spermatogenesis, equal cytoplasm is distributed to, here we have equal cytoplasm distributed to the daughter cells. But in case of oogenesis, here we have differences. It is large secondary oocyte, and here we have first polar body is smaller in size. Right? In case oogenesis, most of the cytoplasm goes to secondary oocyte. Only similarity between first polar body and secondary oocyte have same chromosome number, and the function of first polar body is merely to get rid of the extra chromosome number. Means only we have dysfunction. So secondary oocyte, then another further undergoes second meiotic division, similarly produces to ovum, same, and second polar body, similarly to get rid of the extra chromosome number, right? So these both are the process. Now we have this spermatid, and this is the circular in shape, and this transforms to the next long sperm. Here is sperm we have. Spermatogenesis is the formation of the circular cell. Here is the spermatid. It has the chromosome content is 22 plus X or 22 plus Y. Only the half number of chromosome. It is a haploid cell. Reduces to transform to this linear cell. Here we have head. This sperm has the structure: head, neck, it's a middle piece, and tail, right? So here we have nucleus in this case, in the cell, and mitochondria, Golgi apparatus. So first Golgi apparatus forms the acrosomic cap of the head, and then nucleus forms the main head part of the sperm. This is the neck part, only we have short this. And next is the axial filament. It's a proximal centriole and distal centriole. Proximal centriole transformed to the this ring-like structure, and the distal centriole comes to the annulus part of the middle piece. Here we also have mitochondria. Mitochondria converts to the sheath of the middle piece. And axial filament, in case of cell, produces the tail part. So it's the middle piece of the tail part. Tail actually is used for the movement in case during the process of fertilization we have. So this is the all topic we have discussed today is spermatogenesis, oogenesis, and spermiogenesis. So briefly, spermatogenesis, as we already know, that is the formation of spermatozoa from the spermatogonium, and spermatogonium are produced in the seminiferous of the tubules of the testis, and in case of male. And oogenesis is the formation of ovum from the oogonium, right? And spermiogenesis is the transformation of spermatid to the, it's a circular cell, to the linear cell is a sperm, right? Here we also discussed that two types of cell division we have, mitosis and meiosis. Mitosis only is the just the to make the carbon copy of the parent cell, and is also the equational division, means the chromosome number remains the same after the cell division. And in case of meiosis, we have two categories, first meiotic division and second meiotic division. Similarly, both in the cases spermatogenesis and oogenesis. First meiotic division is the reductional division, means chromosome number reduces to the half, right? Here we also have both cases. And second meiotic division is the equational division, as in case of mitosis, means chromosome number remains the same. And spermiogenesis process describe how circular cell transform to the after the development of main sperm. And this is the process. Thank you.